Okay, what we're going to have a look at here is recombinant DNA or gene splicing. And let's just stop and think about what those words mean. So to recombine means to put a different combination together of DNA, or to gene splice, to splice another gene in. And what we're basically talking about is getting a gene from one organism and inserting it into the DNA of another organism. Now what springs to mind is all these horror Photoshop sort of animals that you might find on YouTube with freakish combinations, and I can assure you that that's not what we're capable of doing in science at all at the moment. What we're dealing with is really just dealing with the DNA of bacteria and looking at plasmids. We do have the capabilities of extracting DNA from the chromosomes of eukaryotic animals and using those genes in the plasmids, but we don't have really the ability to do much manipulation of the actual chromosomes of more complex animals, not to the same degree we can with the plasmids anyway. So what we're really talking about here is identifying a gene or a section of DNA of interest and inserting it into a plasmid. And the way we go about that is we cut the DNA and the plasmid with the same restriction enzyme, so we have the same sticky ends, and we basically encourage those two pieces of DNA to join up. To make that join, we're going to need ligase, but that join can only really occur if both pieces of DNA are being cut with the same restriction enzyme, which will make them complementary. So looking at a bit of an overview of this process, what we're really after is getting lots of these sections of the DNA gene that we want to insert, lots of these plasmids, cutting them both, creating these sticky ends so that they're matched together. And we form these recombinant plasmids. We're hoping that the bacteria will then take up these plasmids, and that's called when they're being transformed. The bacteria have been transformed. These bacteria then reproduce, and we end up with lots of copies of the bacteria, and therefore lots of copies of the plasmid. These bacteria cells will be reading the gene that's been inserted. So if it's a human gene, they'll be reading it in the same code as if it was a human gene. So if it was a gene to make a particular protein, they could be reading that. Now they've had quite a bit of success doing this with a couple of things. One example is the human growth hormone, and they've been able to actually produce this hormone. They've been able to isolate the genes in humans, and they've been able to insert it into a plasmid. So they cut the plasmid with the restriction enzyme. They cut the hormone with the restriction enzyme, and they are able to join them together, insert them in bacteria. The bacteria then transcribe and translate a particular protein, and in this case it'll be the protein for the growth hormone. They've had success with a couple of things. The other one's been insulin, and being able to produce insulin and human growth hormone artificially have been very beneficial. Now, one strategy worth mentioning is sometimes scientists might insert an extra gene besides the one for the growth hormone or the insulin. They might actually insert a gene for antibiotic resistance, and that might just help them isolate the cells who have been transformed, the bacteria cells that have taken up this plasmid. And the way that'll work is there'll be some cells that'll take up the plasmid and other cells that won't. And the way that the scientists try and do this is they try and heat shock the bacteria, and they sometimes have a particular balance of chemicals in the solution, and that'll just encourage the plasmid to move across the cell membrane into the cell. It doesn't happen all the time, but they want to find which cells have taken this plasmid and which ones haven't. So what they do is they put the cells in a nutrient agar treated with ampicillin, which is an antibiotic. And because the cells with the DNA of interest also have this antibiotic resistant gene, only the cells that have the plasmid will survive. All these cells will be toast, and you're going to end up with a colony of bacteria who have been transformed and are able to produce the protein that you're after. Now, scientists have come up with some interesting ways to try and get this manipulated DNA into cells, and in some plants they've been quite successful using bacteriophages to inject the DNA into the plant cells, and likewise in some animals they've used viruses, which, as we know, go to a cell and they inject their DNA in to take over the cell, and that's been a very interesting sort of method to try and get that DNA directly into the cells and be able to act. Alright, so good luck with studying recombinant DNA or gene splicing.